Hello, this is photosynthesis. Here's the equation: 6 carbon dioxides plus 6 H2Os, which is water, plus light energy create C6H12O6, which is glucose, a form of chemical energy, plus 6O2, which is oxygen, just a kind of important byproduct considering we need to live. Now, this light energy comes from the sun. Wow. So here are plants. These are autotrophs. They take light energy from the sun to produce organic compounds from inorganic compounds, which is really important because, these produce the organic compounds that we will eat and stuff for energy. And the light energy comes from the sun. Oh wow! 92 million miles away. Anyway, so it travels all the way to here, and here's the light reactions. Basically, photosynthesis is in two different steps: the light reactions and the Calvin cycle. We'll be starting with the light reactions. Basically, it needs light. Wow. So here's my artistic drawing because I cannot draw. So a photon from the sun hits photosystem 2, the light harvesting complex of the photosystem 2, and a chlorophyll A in the light harvesting complex. Electron is excited, and as it falls, it excites this electron, and the process continues until it hits the P680 chlorophyll A electron, which is then excited up to the primary electron receptor. The P680 stands for the wavelength that this operates best at, so 680 nanometers. This one's 700. So, when this electron is promoted, an electron needs to replace it. So, through a miraculous process, water is split. An electron goes on to P680 to replace it. And I don't know, oxygen is produced, kind of important, along with some H pluses, which are also important. This is in the thylakoid lumen, just remember that. Now, the electron travels down the electron transport chain, which I will talk about, and it passes through the cytochrome complex. Then, so in photosystem 1, a photon hits here, excites an electron on this pigment chlorophyll. It passes energy until it's P700 chlorophyll A, which promotes the electron to the primary electron receptor, and this electron is replaced by the electron from photosystem 2. Now, this electron travels to NADP plus reductase, which turns NADP plus H plus with the electron into NADPH. So this is will be used later as a source of reducing power. Now, the cytochrome complex. So as electrons are falling, basically this is an exergonic process, so energy is released. The cytochrome complex uses this energy to pump H plus from the stroma into the thylakoid lumen. Again, more H plus into the lumen. You'll see why this is important later. Now, to just reiterate, water split into H pluses and oxygen that I need to live, wow. And then these H pluses are again in the lumen. This is very important. Oh yeah, also an H plus is taken out of the stroma to make the NADPH. So again, the stroma is losing H pluses and the lumen is gaining H pluses. So in chemiosmosis, which is the process that produces ATP from an ADP and a phosphate group, the H pluses have an extremely high concentration in the lumen, and the stroma has a low concentration. So H pluses will flow down the concentration gradient through a protein called ATP synthase into the stroma. Now this process is again exergonic as it's favorable high to low. So as it travels through the ATP synthase, the protein will use this energy to combine ADP plus a phosphate group to form ATP, a form of energy for the cell. So basically, the light reactions form ATP, energy, and NADPH, which basically carries electrons to the Calvin cycle. These are the two products of the light reactions, along with oxygen, oh my god, and it's important. Okay. So now we'll talk about the Calvin cycle, which needs no light. So here's an overview basically of photosynthesis again. Here are the light reactions. Now it takes ATP and NADPH, that's what's produced, and it goes into the Calvin cycle. The Calvin cycle will use this to make NADP+, which will be used in the light reactions, and ADP in a phosphate group. And sugar! Wow, that's the whole point. So the first step of the Calvin cycle is carbon fixation. It fixes carbon in the form of carbon dioxide into organic molecules, so basically inorganic compounds into organic compounds. So first, carbon dioxide with the help of the Risco, which is an enzyme, is attached to the 5-carbon sugar ribulose biphosphate. Now, this forms a 6-carbon sugar, but it's very unstable, so immediately splits into 2 3-carbon molecules, which are called 3-phosphoglycerate. This P I totally didn't forget and had to type it in. Yeah, I didn't. And then the second step will be reduction of these two. Reduction, if you didn't know, is adding electrons. So 2 ATP is used. So these 2 ATP become 2 ADP. I made it sad because it lost the phosphate. It's so sad. So this phosphate is attached to the 3-carbon molecule. So now it has 2 phosphates here, 2 phosphates here. And then NADPH, which again was the reducing power thing produced by the light reaction. It gives up an electron to the biphosphoglycerate. And now this biphosphoglycerate loses the phosphate group. And also NADPH is turned into NADP+, which is produced by the Calvin cycle, to become G3P. This is important. This is you'll see. Now, after 3 cycles, we can establish that there were 6 G3P produced, as this one pass made 2 G3Ps, so 6 are produced in 3 cycles. Now, 1 G3P can be let out of cycle, which is the whole point, as this organic compound will be used as the start of metabolic pathways to make glucose and other organic compounds. So basically, carbon was turned into an organic compound. Yay! And then 5 G3Ps will be used for the third step of the Calvin cycle, regeneration. So 5 G3Ps use the energy of 3 ATPs to make 3 5C sugars, which are the ribulose biphosphates. Remember, this was used in the first step, and this was a molecule in which carbon dioxide was incorporated into. So the Calvin cycle, again, makes its own reactants, basically. So 3 ATP is turned into 3 ADP, and they're kind of sad because it lost some, phosphate. But this allows the Calvin cycle to keep going. So to recap, in the Calvin cycle, in 3 cycles, so 3 turns in the Calvin cycle, 9 ATP is used and 6 NADPH is used, but this creates 1 G3P, which is important to start other metabolic pathways to make, glucose, form of energy. And now, this produces ADP and NADP+. However, light reactions will use this to make some more ATP and NADPH for the Calvin cycle. So it's basically one part of photosynthesis makes a reactants of the other one, and vice versa. So that's all. It's really confusing, but maybe a little less now, because if you look at this diagram, it's basically just 1 G3P is made using the Calvin cycle and light reactions. So, yeah. Yay!
Alright, well, hi everyone, and here's our second portion, where we'll be covering a very important process in the cell called aerobic cellular respiration. Now, I'm sure you've heard of cellular respiration, and you know that it provides a lot of power to the body and all the cells, and that it's very important. And you also might be looking at the equation as shown here, and be thinking that, it's sort of the opposite of photosynthesis. You've got a molecule of glucose going in, and oxygen going in, and carbon dioxide coming out, and water coming out, and energy coming out. But really, it's not that these processes are opposite so much as just the reactants and products happen to be switched, and that's actually very useful for plants because they do both photosynthesis and cellular respiration, so they have the reactants and products they need right there for them. And the main important thing with cellular respiration is that you're producing energy. Without the energy produced in this process, definitely humans and most organisms simply wouldn't be able to complete all of the processes that they need to do to survive. So cellular respiration is incredibly important. And as implied by aerobic, it requires oxygen. There are other forms without oxygen, but we'll be focusing on aerobic cellular respiration today. So we have glucose going in, and you might be wondering where this glucose is coming from. Because, we're not plants. Plants are very happy because they have glucose being produced by photosynthesis, and they're doing cellular respiration with that glucose that has been produced. So they've got all they need, really. They've got it going on. But us, we're not so lucky. We don't produce our own glucose. But we do get it from food. And so you'll get glucose from sugars and starches, different carbohydrates. But you can also get some intermediates of the cellular respiration process through other macromolecules, such as fats and proteins. But mainly we'll just be following a glucose molecule as it goes through the process today. And it's extremely important that we have this feeder material, and that's why we need to eat. various things. And so when you're really thinking, food does a lot of things and provides a lot of structural components for your body. So we've got 4 steps here, and it's important to know where they're located. So 4 steps in cellular respiration, we'll go over each of them in detail. Our first one is in the cytosol, and I'm outlining the whole cell here. So that first step is in the cytosol of the cell, outside the mitochondria still. And then the others will be in the organelle known as the mitochondria, which provides power to much of the cell. The second and third are in what is called the matrix of the mitochondria, which is in the inner membrane. It's inside that. And then our fourth step is on the cristae, which is an infolding of the inner membrane. You'll notice that a mitochondria has an inner membrane and an outer membrane, and a matrix space on the very inside, and an intermembrane space in between the inner and outer. It's important to recognize this because we'll be dealing with that very important fourth step on the membrane. So now we have glycolysis, and it is our first step. So you'll notice we start with a 6-carbon molecule, and that would be glucose. So we start with glucose, and the second molecule here, it looks like 5, but it's been split. It's supposed to be 6 as well. And phosphate is not shown, but phosphate has been added to the end. And then we get split into 2 different 3-carbon molecules in the third step, and which still have phosphates on them. And then the phosphates are taken off in the final step. And these 3-carbon molecules are called pyruvates, very important. And this whole process produces a total of 2 ATP and 2 NADH. And how we're getting that NADH is you have NAD+. And NAD+ comes in, and some hydrogen is taken from the glucose. And some of the protons and electrons from hydrogen atoms are added to NAD+ and become NADH. And NADH is going to be essential in later processes because it's an electron carrier. So you have glycolysis, which produces these 2 pyruvate. And then here, this pink molecule in the very front is our pyruvate, and it is being transformed into acetyl coenzyme A. So we're basically taking off the carboxyl group, and we're losing it as CO2. Kind of looks like an HCO2 on the right. It's plus 1 CO2. And we're instead putting on a coenzyme A on top of the 2 remaining carbonates, and that is acetyl CoA. We're also producing 1 NADH, and we're 1 CO2 and 1 NADH for each pyruvate, so this happens a total of twice. That's why you see the ×2 there. And then this acetyl CoA is going to go into the next part. Transition reaction, remember, happens in the matrix of mitochondria. So it's also useful to remember that this NADH will be used later in the 4th step. We'll be adding up the NADH totals later. So now we have our 3rd step, which is the Krebs cycle, otherwise known as the citric acid cycle. So we have on top, we have our acetyl CoA starting. And, you know, the carbons are changed, but it's not quite as important to know the details of this, but basically it goes around. You go from 6-carbon to 5-carbon to 4 to 4, and that last 4 is recycled. And you'll see it's added to that original acetyl CoA, so that you get this 6-carbon. You're not just creating carbons out of thin air or anything. But what's really important, actually, is some of the other byproducts that happen because of these conversions that are going on. You end up having 2 molecules of carbon dioxide that are waste products, and 3 more NADH, 1 FADH2, which is like NADH. It starts as FAD and also gains electrons and attracts hydrogen ions, becomes FADH2, and 1 ATP. And all of this is multiplied by 2 because, remember, 2 acetyl CoAs are going to be going in. So you have all these products, and really what's important to remember that comes out of the Krebs cycle are these NADH and FADH2, because that's really what's going to help us get ATP later on. You have these 2 ATP that are produced in total here, but it's nothing compared to what's coming, and so it's really important to remember the NADH and FADH2. And of course the CO2 as a waste product to account for the carbons. Now a little pause and review. We now have accumulated 4 ATP, which, you know, are somewhat significant, 6 carbon dioxide molecules of waste, and 8 NADH and 2 FADH2 from 1 glucose molecule. Now on this coming slide, you'll have to excuse the red X we have there. Miswrote NADH. That is NADH there. So it's going to be kind of a confusing picture. There's a lot going on. It's happening on the inner membrane of the mitochondria on a cristae infolding. But we'll start with the whole electron transport system, the electron transport chain. So you have NADH over here, right? And what NADH is doing is it's carrying all these electrons, and as is FADH2. You've got this staircase. The staircase represents a sequence of protein complexes. And each of these protein complexes, one the one following the previous is more electronegative than the one before, meaning it wants the electron more than the one before it did. And also, each sequential molecule has more free energy. So NADH is going to go to the top, the first complex, and it's going to drop its electron off onto the electron transport chain. And then the electron is going to go down to the next step, and the next step, and the next step, in a series of redox reactions. Remember that reduction is gaining an electron, and oxidation is losing. So each protein complex is gaining one, then losing one, then gaining one, then losing one. And so eventually, these electrons are going to go down and be accepted finally by O2, which, if you'll remember, was a reactant of the entire cellular respiration process. This is where oxidative phosphorylation is going to come in, because we've got all these redox reactions in O2. So you'll notice me circling these parts. The O2 receives the electrons and joins with the hydrogen ion and creates a byproduct of water, which is a waste product. So we have all this going on, and FADH2 is also involved. It's just dropping electrons off at a lower level. So it's not quite as much energy is being released by these redox reactions. So you might be wondering, how would we get ATP from this whole process? How is this related, this staircase? Well, we're releasing energy because it's an exergonic reaction happening as the electron keeps moving down the chain. And these H+ ions that you'll find on the inside can move through these protein complexes out into the inner membrane space in the mitochondria. And so when energy is released from this electron transport chain, these H+ ions will be moving out into the inner membrane space, and because a pump is being created. Now this creates an H+ gradient, which allows for chemiosmosis, which means that a gradient can do work for the cell. So these H+ ions are going to want to move back in because of their higher concentration on the outside and lower concentration on the inside. The only way for them to go back in is through this blue complex here, and this is called ATP synthase, as you'll remember from photosynthesis. Now an H+ ion goes in and basically goes around this ring here, around the ATP synthase. And when this ring turns, it turns a sort of rod-like structure in the center of the ATP synthase, and that rod turning is actually catalyzing the synthesis of ATP, joining ADP, adenosine diphosphate, and a phosphate group together to make ATP. And so when these hydrogen ions are coming in because they want to equalize the gradient, they're actually powering this ATP synthesis. So even though it might not seem like everything's connected, it really is, because the electrons falling are helping the hydrogen ions go out and then come back in through the ATP synthase and creating the ATP. So some bookkeeping. We have our little proportion. So 1 NADH is usually about equal to 3 ATP when the process is said and done. So since we started with 8 NADH, that equates to about 24 ATP. And 1 FADH2, since it is dropped off lower in the process, only produces about 2 ATP, so our 2 FADH2 are about 4 ATP. It's for a total of 28, plus our 4 from the beginning of glycolysis in the Krebs cycle. It's about 32 ATP total per glucose molecule. And so you might be wondering why it's not exactly ATP. Well, it's not an exact number because this electron transport chain is not directly coupled to this ATP synthesis. The NADH and FADH2 dropping off electrons is creating this this pumping action outside, but that's not necessarily proportional. How many electrons are dropped off and how many H+ are pumped out. And also this proton motive force, this chemiosmosis, is also used for other work. So you might not always get that 32 ATP, sometimes maybe a little more. But in general, it's very important just to have this in general because the cell needs all this energy to be able to work. So we have about 32. And remember that the CO2 that was created in the process and the H2O at the very end is just waste. And funnily enough, we actually exhale the water along with carbon dioxide. Plants are happy because they can use that carbon dioxide for photosynthesis. And so why is cellular respiration important? Well, it's important to remember that ATP is energy for the cell. We wouldn't be able to do anything without it. If you think about any reaction that we've studied so far in the year, there's really nothing that can be done without ATP if it's an endergonic reaction. And just in general, there are a lot of very essential processes that are driven by it. So the fact that cellular respiration creates a lot of it is frankly awesome, and we should be very thankful that aerobic cellular respiration works for us. Fermentation simply doesn't produce as much, and that's why we don't do a lot of it. So aerobic cellular respiration is definitely the way to go. And more ATP equals happy cell, and happy cell equals happy life. I hope you've enjoyed this presentation and that you've learned a little bit more about cellular respiration and what it means. And If you have any questions, you can leave in the comments below. Thank you for listening to our podcast. This is Karen Boone with Brian So, and please look in the credits for citations. Thank you very much.